Obviously, it is your business.
Wait for another 1 minute or 2 minutes to join the participant. Okay.
That it's a collective effort, but the blessings, your blessings are always there with us. I believe personally.
It's not blessing. You can say, yes, my good wishes are always there with you. Of course, it is your effort, tremendous amount of workload and capability.
I think, Ritu, we should start.
Yes, sir. Okay, sir. Okay.
This is all yours. Thank you.
Okay. Good morning to all. Myself, Ritu Khara, Assistant Professor, JISU University. I would like to welcome you all in second session of last day of FDP entitled Recent Trend in Pharmaceutical and Biochemical Research, organized by JISU University. It is my great pleasure to introduce our eminent speaker, Professor Dr. Biswajit Mukherjee. Professor Mukherjee is an outstanding researcher and eminent educationalist. Now, Dr. Mukherjee is professor in pharmaceutics and former HOD in the Department of Pharmaceutical Technology, Jadavpur University, Kolkata. Professor Mukherjee is a former DART fellow, ex-guest scientist, German Cancer Research Center, Germany. He is also former visiting fellow of the School of Pharmacy, University of London, UK, and the former Indo-Hungarian Education Exchange Fellow, Hungary. He has been working on novel drug deliveries, pharmaceutical, particularly transdermal patches, liposomal, and mucosomal formulations. He has supervised many projects of different funding agencies such as UGC, CSIR, ICMR, DST, DBT, etc., and publishing his work in many national and international journals of repute. Having more than 23 years of experience in teaching, he has received many awards during his journey, one being the UGC Research Award in 2019 and has few patents in process. This is the very brief introduction regarding our eminent speaker. I would like to request Professor Mukherjee sir, if you please proceed your session sir, please start. The stage is yours, sir.
So first of all, I'd like to convey my sincere thanks and gratitude for your nice introduction. So Dr. Khara has introduced me quite exaggeratedly. And then also I like to convey my sincere thanks and gratitude to the principal and all other, faculties and all the persons who are organizing this particular program. And of course, the participants, those who are here to make it successful. I understand it is the wee hours. It is the last day, second session, possibly another 2 sessions are there in the second hour, another 1 session. So it's wee hours. You have already, filled up with, you have completed the nearly filling up of your bottle of knowledge in this session. So maybe some portion is remaining, and whatever I will say, if you can gain something from it, it will be beneficial for us, particularly for the organizers who has the intention to organize this program. So without wasting time, I will go for the lecture. I need, permission for slideshow.
Yes,
okay.
Sir. Yes,
yes, it's already given. Okay. So, screen this one, share. So I can now go for full slide. Yes, and
perfect, sir.
You can see it, right?
Right.
Okay. So this area particularly I want to discuss. Basically, the targeted drug delivery with the nanosized products, nanosized drug. Products means actually basically, formulations are also drug products. So nanosized drug delivery system, we understand that the vehicle, that the carrier, you want to carry something from one place to the other, that is the carrier. And this carrier like train, tram, bus, truck, taxi, boats, ship, they're all carriers. They carry goods, they carry human beings. So here our carrier will carry drug. But the difference between this carrier and that carrier, there are many common vehicle, common carrier which can carry drugs also, huge amount of drug they carry. But here the difference is they're not only carrying drug, they carry drug in a nano-size form. This is the most important thing. They're not very, very large size, a very, very small size, nano size. And nano size, you understand, submicron size. Second important issue is you're using this drug carrier to deliver drug into the system in our body. That is more important, that in different tissues, different, physiological parts of our body. But not as such that wherever I want to deliver, I am delivering it. It is not like that. In a very specific manner, we want to deliver. That is what we call targeted. Targeted terms means actually you want to deliver in a particular location. That we want to mean the targeted. So targeted delivery system. And if it is targeted to a particular location, obviously it must have better therapeutic potential. Let us see how it can work and really if they have better therapeutic potential or not. Whatever I have said so far, this is the, you can say, picturesque demonstration. This is first one, you can see the target, you are just delivering in a particular location. Second is carrier, but the carrier is nano-sized vehicle. And the third thing is, yes, your target is the cells. Your target is the cellular organelles. So you are giving the drug in a very, very precise location in our body. That is the main important thing. We have to understand really what is nano size, submicron size. We understand submicron size. Can you see it? No, we cannot see it. It is not possible to see it. You have to take the very sophisticated instrumentation, facilities to see it. That is important. So you can see here you can see it is visible. Maybe here, not much, but here it is. See, its size is 100 micron. 100 micron is thickness. Bacteria, even up to 10 micron, 1 micron, they are not nano size. Bacteria you cannot see by naked eyes. You have to take the advantage of some scientific instrument, microscope. But virus, you cannot see it even by ordinary light microscope. You have to take further sophisticated microscope. What you call is electron microscope. You see, virus size is 40 to 100 nanometer. That is the most important thing. You must remember this size, 40 to 100 nanometer. Taking this advantage, they don't need any receptor to go inside your cell. They can permeate inside the cell, taking the advantage of their size. So this size can do many things. Protein, they can enter inside the cell. Size is very, very small, 10 nanometer. Carbon nanotube, it's very small. Even it is smaller, 1 to 2 nanometer. Atom, further smaller atom you cannot see at all. and it is 0.1 nanometer. So you understand it is rather, I should say, in picometer. So 0.1 nanometer is actually 100 picometer. So you understand that these sizes are very, very small size. And this small size, why and how it is important? Small size has enormous potential. I say that, their drug, because of the large size, they're capable to do many things, but they cannot enter inside the cells. Several molecules were discovered and they're rejected. Where? Because they're unable to enter inside the cells to work. But here, if you bring it into nano size, you see they're very specific and they can enter inside the cells. Common advantages are there. I am not going to discuss about these things. These are all bookish information. Like if you convert it to a smaller size, surface area will increase. I am not going to say. I am just going to say very important areas. If you convert any materials into smaller size, in nano size, when you are going to reach, you see, many cases you will get a surface, additional surface properties like supermagnetism. That means, which is not magnet at all, it will have the property of magnetism, which cannot enter inside the cell, that will enter inside the cells. So many such, advantages are there rather than the common advantages which is stipulated here. Chemical properties changes, physical properties changes, lots of physical properties, lots of chemical properties, are changed actually. So here interesting part of the story is, I think those who are entering, I asked Ritu to admit them because that will be nice for me. Now you see, lots of changes are there and some changes are really miraculous. You see, gold ring. Many of you have gold rings in your hand. Although I don't have, but you have, you people are rich people. So you see, this gold ring, if you want to dissolve gold ring, what do you need? You need aquaresia. 3:1 concentrated hydrochloric acid and nitric acid mixture. You have studied it in class 7, 8, or maybe 9th standard. I don't remember, but in a very lower class we learned it. 3:1 hydrochloric acid, nitric acid mixture. Otherwise, this will never get dissolved. You see, if you convert this gold into nano size, they will be dissolved in water. And you see, you will get different solution, different colored solution, depending on their size, no particular color. So a gold even can be dissolved just simply because of its size, nano size. So you understand the importance of this nano size. Of course, if you say whether any disadvantage or not, maybe everything has advantage and disadvantages. Any drug if you use has advantage or as advantages, equally they have disadvantages. Even any material you use has advantages and including water, including oxygen. Nothing is, free from anything. Even oxygen is toxic to our body. Water is toxic to our body. Toxicity depends on how much you are consuming. Anything you consume over than the desired amount, that causes toxicity. So consumption of oxygen more than the desired one, it will cause systemic toxicity. Consumption of huge amount of water will cause toxicity. So remember that within the limit if we take, everything is fine for us. So I'm not going to discuss about this toxicity or other areas. Of course, everything you have to make in a balance. Maybe someday, if we have some topics to discuss, we will discuss on that. Then So important thing is you understand the importance of nanosize. Second is the targeting approach. Here, targeting approach could be two types. One is conventional and the other is non-conventional. Conventional means, that means like you want to drop a missile somewhere, you are tagging with a very, what you call the targeting ligand, it will bring it to a specific location. This is called targeting, direct targeting, conventional targeting. That means you are giving something to target. And not, non-conventional means it is not that you are tagging something like, which will direct it to a particular location, but you are formulating in such a way that it will go for a specific target. So this is, what you call is the non-conventional targeting. So in this area we will discuss. Now, most important issue is we are going to discuss some approaches of targeting. Here is one of our latest work I want to discuss. Here targeting is done, of course, nanocarrier of liver cancer cell. We want to target the liver cancer cell. Hepatocellular carcinoma is nothing but cancers in hepatocyte, liver cancer. Liver cancer means liver has different cells. It is not these hepatocytes alone. There are erythrocytes cells, there are other cells, but you see, major cells of the liver is liver parenchyma, and liver parenchyma is basically, called hepatocyte. So liver parenchymal cell or hepatocyte, when forms cancer, that is a major form of cancer in liver, because that is a major, constitutive cells. And when they form cancer, we say hepatocellular carcinoma. And hepatocellular carcinoma, this is actually a very, very fatal disease, bad disease till date. So it has no good treatment because you see, you have to remove the liver, or maybe the liver may be transplanted, but no guarantee that after the transplantation of liver, people may survive. Because first of all, getting liver is always difficult. Second thing is the acceptance of the organ is very often very little. And, thirdly, partial hepatectomy will never work. And the drugs which are available, they're very toxic. So anyway, this is how we wanted to target liver carcinoma cells. And, the nanocarrier, of course, is the approach. And the drug is paclitaxel, which is not mentioned here on the title. But how we are targeting? Here it's a direct approach. We are targeting with aptamer. Aptamer, this is your formulation, and then the aptamer is conjugated on it, and aptamer will bring it to the right cells. So that is the main approach. Now you see, here I want to first give you the concept of aptamer. What are aptamer basically? Aptamers are the nucleic acid sequence. That is DNA sequences, mostly DNA sequences. And it's a small fragment of Nick DNA. Nick, you understand, single-stranded open DNA. So it is a strand of DNA, single-stranded DNA, normally it is. Question is, single-stranded DNA, if we just give it to the system, will it survive? Answer is no. It cannot survive because you see, in system, lots of enzymes are there, DNAs, RNAs, enzymes. So DNA's enzymes, they will split it, destroy it, and it will never be saved. DNA will be clipped, destroyed. Then what you can do? You have to modify it so that DNA's enzyme cannot work. Where can you modify it? You cannot change the bases. It is never possible. If you change the bases, it will be a perfect mismatch. Adenine, thymine, cytosine, guanine, they are the matching. Adenine and thymine, they're connected with double band. Cytosine and guanine, they are connected with triple bonds. And you cannot connect cytosine with thymine or adenine with guanine. It is not possible. So any mismatch means it will be destroyed immediately by the DNAs. So you cannot change the bases. Then you can change the sugar. It is not possible. If you change the sugar, like DNA deoxyribose sugar, RNA ribose sugar, the characteristics of the nucleic acid molecule will change. It is never possible. You cannot change the sugar moieties also. Then what can you change? You can change only at the phosphodiester linkage. The sugar moieties are connected with phosphate group, one phosphorus and with 4 oxygens. We say it is phosphodiester linkage. So here if we can modify, then DNA's enzyme will not be able to work. Why? Because DNA's enzyme actually, they try to split this particular area. They don't have eyes. They have the scissoring capability, or they have the scissoring capability in terms of, destroying this particular sequence. But when you are modifying it, see in the picture, you are modified. Here you see when you are modifying it with this oxygen by replacing some elements, here particularly we use sulfur. Sulfur, it is called phosphorothioate. Phosphorothioate. So phosphorothioate linkage, when you are incorporating between this, replacing one oxygen, it is not possible for the DNA's enzyme to, recognize that. As a result of that, this single-stranded DNA survives in your blood. So it can work efficiently because it will not be destroyed. Question is, any single-stranded DNA, small sequence, obviously you cannot give a very large sequence. Normally there are 30, 40 bases. So in such small sequence, is it that any DNA sequence is an aptamer? Answer is no. Aptamers are very, very specific sequences. What specificity is there? Why other DNA, nick DNA of this 30, 40 bases are not aptamer? What is the difference between an aptamer and the another single-stranded similar base number sequence? The difference is aptamers are those particular sequences which can form a 3-dimensional structure. You understand 3-dimensional structure. So because of that 3-dimensional structure, they can bind with some specific proteins. It is not that all 3-dimensional structure formed by DNA can form. First of all nick DNA cannot form 3-dimensional structure. Second is some specific nick DNAs can form 3-dimensional structure, which we call aptamer. Third, all aptamer cannot bind with protein. So you have to find out which of these 3D structures can bind with the proteins. So people are working on that. Many people are working on that. First of all, with this, what you call this computer homology model, they decide that which are the possible aptamers, and then people work on that. They find that out of maybe 100, 4 or 5 are actually working, the remaining are not working. So out of these 4 or 5, then again they select which one is more efficient, and then they try to find out which protein there can be the target. So it's a complicated thing to find out with the aptamer. So we have selected such one aptamer, and conjugated with the nanoparticle, and that is our ligand to target to the liver cancer cells. Now, let us go for the actual results. You see, this is SCM, scanning electron microscope, TEM, AFM. So here basically you can visualize the particles, visualize these particles, and by different ways. So, you know, this surface has aptamer. Drugs are, you know, scattered inside these particles. They're not homogeneously distributed, but scattered, and size distribution and the shape is more or less uniform. And then we worked with 5 different aptamers and 2 different proteins, mainly, you know, the apotransferrin and also galactosamine proteins we worked with. Along with that, we have taken 5 different aptamers. We named it L1, L2, L3, L4, L5. And out of that, we found that 2 aptamers work very nicely, L2 and L5. And their data actually you have produced here. You see, here in the well, you know, the nanoparticle conjugated with aptamers are there. Because they are heavier, they could not move. But free aptamers, you see they are here. We have different size, 2 different sizes of free aptamers we have used. One is conjugated with this and the other is conjugated with this. And these are the DNA ladder, just to understand that sizes are same or not, etc. So we found that conjugation is perfect. So aptamer is conjugated, free aptamers having the proper size, and aptamer conjugated nanoparticles are in the well. Then the major important thing is we want to see whether they are taken up by the cells. You are prepared, fantastic. You have prepared the nanoparticle, here it is conjugated with aptamer, it is very nice. But the most important thing you have to see whether they are going inside the cells or not. This is important. And whether they're going inside the cells or not, if you want to see, for that, you know, that cellular uptake study you have to do, and that we have done with 2 different types of cells. One is, you know, HepG2 cells, and the other is HUH cells. These 2 cells, HUH7 cells and HepG2 cells, they are all, you know, basically what you call is cancerous cells, cancerous cells. So, cancerous cells. Again, we see whenever you are going for any in vitro studies, whenever you have, just a minute, somebody is disturbing. Yeah. So what I was telling you, sorry, because somebody was shouting here. Shouting means, you know, they're selling their products. We can say they're periwala, so we cannot stop them. But anyway, he is disturbing me. So what is important here? Once you have prepared it, you know, you have to see whether they are going inside the cells or not. And there, you know, at least you have to take 1 normal cells and more than 1 cancerous cells. So we have taken up 2 different cancerous cells. Both of them are hepatocarcinoma cells. One is HepG2 and the other is HUH7. And what findings we have got, very interesting findings we have got, is these are all normal control results. And you see, in different hours, 1 hour and 4 hour study, 1 hour and 4 hour studies, and HUH, this one HUH, and this one is AB is your FG2 cells, and you see, this is the normal finding. Normal, with the normal, you see changes are there. Changes are there. And if you go for L5, so almost nearly 3 times higher uptake is there. At 1 hour and at 4 hours, this uptake goes to 2400, that means 6 times. 6 times. Same is true here also in HOH. So you see the values comparing to this, you can say L2 is having also similar, but not exactly similar, little less uptake comparing to the others. So we understand that these products are going inside the cells. And then we have gone for these IC50 studies, and of course the cell inhibition, cell viability, we have investigated. And here also you can see L5 has the lowest molar concentration found to be effective. So since it is going more, of course it will work better at a lesser concentration. So that proves the thing. And yes, in the other 2 cells, normal cells, the viability is more, inhibition percentage is less. You see, it is not killing the cells in normal case, but 50% killing at this concentration is there in case of the cancerous cells. So other 2 cells we have taken is Chang liver and WRL68. WRL68 and Chang liver, they're basically, you know, what you call is this normal cells. Then if we go for the cellular uptake in Chang liver and the WRL68, these are the findings. Very interesting finding. You know that all these studies have been done with flow cytometer, FACS, what you call FACS, flow cytometer. So you understand this has great implication. These results are very, very, you know, authentic. So the control results, you see control results. Now you go for L5. You see more or less same as control. More or less same as control. That means, you see, the same formulation which is working very, very nicely, or rather efficiently, I should say, in cancer cells, they are unable to work in normal cells. Normal cells, that means, are not being affected with the formulation. What is the reason? Possibly they are unable to enter inside the normal cells as it is there inside the cancer cell. And that this data shows that thing. This data shows that thing, that if you compare with the control, you see in the normal cell uptake is similar like control. Whereas if you go for these cancerous cells, you see uptake is much different compared to control. Uptake is about 6 times higher. So that is a difference, you know, between these 2 results. So this is very encouraging in that sense that this is not going inside the normal cell, and that's why it is not affecting normal cells much, but it is going inside the cancer cell much more in terms of, or internalization in the cancer cells is much more compared to the normal cells. That's why it is effective, more effective. So that's the finding, encouraging finding, what we wanted to get, that this targeting means it will go to the cancer cells, it will not go to the normal cells. And that is more or less achieved by this finding. This is the first initial findings. Then we have gone for other secondary studies. You see, when you are just going for a study, if you have a single finding, nobody will accept it. Nobody will believe it. You need at least 1 more study or maybe more studies to substantiate that, yes, what findings you have shown, it is correct. Because in a single finding, people may not believe it. So that's why, you know, that secondary finding, supportive finding is required. Here you see we have compared all the different formulations, including this protein conjugated formulation, marketed formulation, and all these different formulation. Free drug also, and this is the entire comparative studies in HepG2 and HUH. A is HepG2 cells, first two, and HUH is the B category cells, the second. So this is also, you see, findings is also there. This is your control, and this is your aptamer conjugated nanoparticle L5. Here also this is L5, this is control, and free drug marketed formulation, and then this only nanoparticle, this is aptamer L5 conjugated nanoparticle, L1, this is I think L2, L1, L4, this is protein conjugated. So different nanoparticles. This is what we have studied. This is cell cycle analysis. You know, paclitaxel, what does it do? It actually blocks the cell division mitotic process. So when cell forms spindles before that is arrested by paclitaxel. So this is a process by which drug works. So if your drug goes inside, if your drug goes more inside, obviously it will show its arrest at the mitotic division. You know, cell division cycles. Cell division cycle is, what do we say? G1, that is also G0, but G0 is not considered, well, it's very for specific cell. So G1, S, G2, M2, or G2M, whatever. So you see what is there. G1 is what? G1 is after the cell division growth. After the cell division growth phase is called G1. S, again for the next cell division, the DNA synthesis and other things, DNA enhancement of the synthesis of that is S phase. Then G2, not only DNA, now the proteins, other RNAs and other proteins, they are preparing for the cell division. And M, basically, what is called mitotic. So mitotic process. You see here in L5, G2M phase 55%. Compared to any other, you see, this is the highest one. You can check from on this data. The highest one, 55%, 55 arresting at G2M. That means at the mitotic cell division. And that proves, yes, your data is correct. Uptake is there means it is also arresting the mitotic cell division. That means it is controlling the cell division when you are treating the cells with L5. You have all of these two, A and B, they're cancerous cells. And you have given different treatments. You see how difference is there. Just see the free drug. Free drug is just 3.1% arresting. That means it is not going inside the cell really. And, or going, but it is taking long time to work. Or it is going very less. Whereas you see, commercial formulation arresting that phase only 11% at the same time point, whereas it is 55%. These are all very, very authentic data, that it is not that you cannot change, all with the help of facts. So very, very reliable. Then another important study we have done. This is, what you call tunnel assay. Tunnel assay, it is basically, ApoB DNA fragmentation assay. That means, you see, how much DNA is fragmented. In this case, if you just see the results, you see control is 3,987, whereas it's 86,000 in L5 in both the cells, A and B group, that is HepG2 and HUA. You see, control is 4,000 and it is 86,000. So nowhere any other result is so close, even not L2. L2 is here, you see, 57,000. Although, of course, this is much more than control, but nowhere it is close to 50. This is 86,000, and this is 86,000 again, very close. But here is the L2 is only 51,000. So this is very interesting. In two different cell lines, HepG2 and HUA, both of them are cancerous cell lines. And here you see, DNA fragmentation is much, much more when you are treating it. That means the cell is dying, cell is getting destroyed. So this is a very interesting finding. So we now want to see what mechanism it is actually adopting to the cellular destruction. Here, let me tell you what I want to mean the mechanism. There are two types of mechanism grossly which can affect the process of dying. One is, there are many other processes also, you can say three also, but more predominantly two processes. One is apoptosis and the other is necrosis. If necrosis occurs, without apoptosis, if necrosis occurs, that indicates this is very toxic. So this is a very unsuccessful effort of your research. If you see the cell has necrotic property or it is undergoing necrosis, that means you can say your research is finished. You have to stop the research. You have to either reduce the concentration, or you have to change the drug, or you have to change the formulation. That means it's a big upset. So at this stage, we understand cell is dying. After this is fine. It is not affecting normal cell. It is fine. It is affecting cancer cell. It is fine. But here the main approach is how it is killing the cancer cell. Necrosis or apoptosis. If necrosis, our research is finished here. If apoptosis, our research begins or starts from here. So you see, we studied the apoptosis process. Apoptosis, that we have done it both qualitatively and quantitatively, because you have to understand that what you are saying, what you are doing is correct. Just single data may not be sufficient. So you see what we did, this is for the apoptotic study. Basically, apoptosis is done with this acridine orange staining and with this ethidium bromide. So normal case, what happens? In normal cell, it is green. In early apoptotic cell, it is green. Late apoptotic cells, orange to red. And necrotic cells, red. So now I will differentiate. So this is a coloring agent, acridine orange and ethidium bromide. But the color of the cells changes. I repeat once more, normal, green. Early apoptosis, green. Late apoptotic cells, orange to red. Necrosis, red. Then where is the difference? How should you differentiate them? There are procedures. What is the procedure? In normal cells, it is green and the nuclear structure is maintained. And of course, it is much different from the early apoptotic cell. Early apoptotic cell, what happens, that the cells, membrane, blabbing will be there, membrane chain alteration. You know that it will have the nuclear having the chromatid body, chromatins at the perinuclear regions. You will get the apoptotic bodies. You will get, the shrinkage of the nucleus. So many such phenomena normally occurs in case of this, what you call this, early apoptotic cells. In the late apoptotic cells, similar phenomenon you will find, but color is changed. Necrosis is morphologically the same like normal cells. Same like normal cells. But the difference is the color is red. So the difference between necrosis and the apoptotic cell is morphologically no alteration of this necrotic cells with the normal cells. But in case of this apoptotic, late apoptotic cells, they have lots of changes. You know what I said, that nuclear shrinkage, sometimes, the chromatid body formation, this chromatin, the peripheral perinuclear region, lots of, what you call this apoptotic body. So you can see here this different formulations and the apoptotic appearances. First one is the cancer control cells. You see there are many early apoptotic cells also, because this is cancer sometimes, but which is very predominant in case of this commercial formulation. Commercial formulation, you see lots of early apoptotic cells are there, you can see. But if you go for this nanoparticle formulation, there also, along with the early apoptotic, late apoptotic cells are there, at least maybe 30%, which is much more in L5. Late apoptotic cells are nearly, you can say, about 80%. Same is true in L2 and L1, maybe it is also there, but little bit late. So you understand that mainly apoptotic cells are there. And this is also another, cell lines. Here also you can say in Hua cells, control, and along with that you see the different formulation. There are similar type of changes are there, but maximum you will get in L5, L2, and followed by others. So you see the apoptosis is there. Now these two cells, HepG2 and Hua, whatever I have shown you, they are given in the single slide so that you can understand the changes. How do they look like due to the variation of the cells? That at a glance you can get the vision of them. So you can just see that the changes are more or less similar. Now, if we go for another study, that is just to see the mitochondrial membrane potential. Mitochondrial membrane potential we want to investigate. And normally, mitochondrial membrane potential, it is normally, we do it with this, what you call JISU1. JISU1 is monomer. JISU1 monomer, when it goes inside the cell, if the alteration of the mitochondrial membrane takes place, they aggregate and they form green color to red color. So normally this is happened, and the ratio is important here. Based on that ratio, we consider, you see, the P3, P4, there are two, I do not know whether you can see it or not, P3, P4. And the P3 portion is the live cells, P4 is the apoptotic cells. So you see, if you compare, you will find that apoptotic cell is much more, 68% in case of L5, which is supportive of all the earlier experiments. And this is also there in other cases, also in the other at 4-hour study also. Then if you go for the C and D, that is HUH cell, there also another interesting part is there, that is in the different quadrant, what type of cells are there. You see here, when you are getting four quadrants, these quadrants actually here just to indicate the changes. I just want to tell you first, so this is a control one. This is live cells, live. This is second quadrant, you can say early apoptotic cell. This is late apoptotic cell, and this is, you see, necrosis. So if you just consider control cell, maximum 98% of the cells are in the normal region. But if you go for, anyway, it is getting inside behind this picture, but let's see, I can. So yeah, so and here you see L5 here. 19% is live, then 58% early apoptotic, 28% late apoptotic. This is necrosis. So you see, in every case, live, then early to late apoptotic, every case you are getting. So that indicates this is absolutely going to a right direction. They're going to a right direction. That means early apoptotic to late apoptotic phase. And this is very encouraging that necrosis is not taking place. Now, you see, this is, again, this is also done by FACS, and this study is related to Ross generation, level of reactive oxygen species. Reactive oxygen species generation, Ross generation. And this is also done with first one, A group means it is HepG2, and this is another cancer cells, HUH. Here you see Ross generation, if you compare with the control, it is 44,000 something. Again, here you see LNL5, 44,000. So it is comparing to any other formulation, you see this is exaggeratedly high. That means you see lots of reactive oxygen species are generated in cancer cells, and that is what you call is maybe one of the major cause of this apoptosis or inducing apoptosis. You just go for these confocal microscopic images of the normal cells, Chang liver and WRL8. You see, normal cells, just see how much impact it is. Normal cell control, and this is L5 treated. This is normal cell control who are L5 treated. In both the cases, you see no change compared to control. Same is true with the nanoparticles, paclitaxel nanoparticle without even the treatment, without even ligation. Same pictures are there, you see, no changes. But when you are treating with marketed formulation, how much normal cells are dying? And you see this death is similar structure of this, more or less similar structure of this normal cell. That is necrotic death. This is causing lots of, killing of the normal cells at the, but for the treatment of the commercial formulation. See, so how much it is dangerous to take the formulation which is not targeted. It is killing your normal cells. So many normal cells are affected. See, in both the cases, they're not the cancerous cells. You have done it with Chang liver and WRL8. So you see, the normal cells or treatment of this protein conjugated nanoparticle, aptamer L1. So all the cases you see it causes toxicity, which is not there in L5. So what we understand at this stage is L5 is having enormous potential to target the liver cancer cells without affecting the normal cells. So now, once we have got it, we want to investigate it in the animal model, that is in vitro to in vivo. And you see, we have taken different animal models. Animal models, basically, 8 groups we have taken: normal control, carcinogen control, that is cancer, liver cancers, then carcinogen control with the treatment, then carcinogen control with the treatment. There are 2 treatments we did with L5, L2, etc. And then also the normal animals treated with the, what you call is, the formulation. Now, if we go for the liver of the cancerous animals, what you can see, different types of changes you will find. First picture, hematoxylin, you see there are lots of ground glass appearance, ground glass focal lesions are there. In the second case, you will get the basophilic focal lesion. And third one, they're like void, they're not void actually, they're cells having pointed nucleus at the periphery. And basically, what you call, they're called spongiostis hepatis. So you see, all of them are, you can say, hepatic altered focal lesion, or we can say hepatic altered foci, or HAF, HAF. So once the treatment is there, you see there are changes in the liver, and basically changes are very satisfactory when you are giving a treatment of this L5. It is the most satisfactory result. We have often, you can see it here, and the changes, how much being modified, you see it is more or less close to the normal cells. And then you see, this is the counting, what I have shown you. This is the counting that is in the animals per square centimeter area, how much lesions are there based on that. So that also reflects that smaller lesion is comparatively more in case of this L5, whereas larger lesion, like greater than 3 millimeter lesions, are there more in case of the other cases. So that means it is definitely delaying the cancer process. We have also investigated the other apoptotic marker and anti-apoptotic marker also, and we compared, and that they actually are supportive of our earlier finding that yes, apoptosis is going on in the system. Then pharmacokinetic data of the formulation. Here also we have investigated in different cases, and you see what we found, although the picture is not very, I think, clear, but you see free drug and the formulation, they have actually free drug ends here, maybe by 36 hours it is it is eliminating totally from blood, whereas it remains more than 72 hours. I think it's very close, it will remain in the system. So that means obviously this is a formulation, a nanoparticle, so it will be there in the system longer, and its half-life will be more, so it will provide some sustained action also. This is the liver, normal liver structure after the treatment of L5. See, no much changes is there in case of these animals treated with this formulation without having cancer. So the animals without having cancer, when treated with this formulation, it doesn't cause much necrosis of these liver cells or hepatocytes, no much damage in the liver that way if you compare these two pictures. So that indicates no much effect of the normal hepatocytes when the formulations is given. And this is also, the other biochemical parameters we have investigated, common parameter, what you call which reflects the liver toxicity, that is SGOT, SGPT, alkaline phosphatase, etc. So there also you can see the visible changes is no much toxicity it is producing, both L5 and L2 compared to control. And in different number of days is also we have studied and see data reflects that changes are not there. That means when animals are treated with the normal, the animals treated with this formulation, they're not causing toxicity up to a treatment period of 28 days. So this is how we have now reached the findings, or reached that definitely L5 has the potential to control the neoplastic hepatocytes, and it is not causing toxicity in normal hepatocytes, which is actually the you can say the result indicates directly the target is successful. Now question is here, although this part I have not done, it's done by my colleague, he has the expertise in it. Using this homology model, we wanted to know, okay, this aptamer is successfully working, both L2 and L5, maybe L5 is better, but L2 is also capable of working. Now we want to know that which protein it binds, whether that protein is at all exist on the liver cancer cells or not, whether they're existing in the normal cells or not, and what are those proteins actually. With this homology model, we investigated, and L5 and L2, the proteins, neoplastic hepatocytes, they produce these proteins, Hitchcock protein 70 and TAC 72. So these proteins, we believe that this aptamer binds with these proteins, which is available on hepatocytes, cancerous hepatocytes, and then, they binds and they go inside the cells, and that's why they're so successful to reach in the cancer cells, but they are not very successful to reach in the normal cells. So I want to know, yes, I want to know how much time is there.
Sir, please, you take no time.
Now you tell me, accordingly I will continue.
Sir, the session will be concluded at 1 p.m. including the interaction.
Okay, so I may take few more minutes. Okay.
Sure.
So whatever I have shown so far, this is related to aptamer conjugated nanoparticles for drug targeting. Here, let me tell you that aptamer conjugated nanoparticle for drug targeting, basically, aptamers are called, what you call this, chemical antibody. They're so specific to a particular protein. Chemical monoclonal antibody. Anyway, this is another study I want to show you. This is a target. This is also non-conventional. This is, you are targeting in the lungs. Basically, the lungs, what happens, lungs when it has fungal infection, particularly it happens in the immunocompromised patient. So when lung has fungal infection, it is very difficult to get recovery. Why? Because you see the drugs are very competent. Voriconazole, itraconazole, ketoconazole. So they are all very competent drugs. In vitro, you see, they are very successful to control the, what you call this, fungus. But when it is the case of lungs, what happens, why it is unsuccessful? Because lung has a very high blood turnover, very high. As a result of that, persistent maintenance of drug concentration in lung is practically impossible. That results in, for that, huge amount of drug concentration or a huge amount of dose you have to give. That is not possible. It will be very toxic. Secondly, even if you can do it for some days or some weeks, you have to stop after that. But this cancer, the fungus spore, they remain at the lower part or deeper inside the lungs, and recurrence of the disease occurs. And thereby, people die because of this, lung infection with this fungus. That's why till date, there is no survival if you have fungal infection in lungs. So for that, what we wanted to do, although this investigation was done in animals, but we wanted to see whether if we keep the drugs in the nanoparticulate form containing voriconazole, and whether it will remain in the alveoli, and if it can produce the drug release in the alveoli for longer period, you don't need to maintain this, blood level. It will work locally, and it could be a successful approach. You can additionally give, if you another treatment whenever it is required through blood also, but it may not be required also. So our approach was simply, we use these two devices. For this one is for the treatment of animals. We have developed it by ourselves. 46 tubes are there, and there is two barriers inside is nose-only apparatus. Noses are there of the animals, and this is, we have prepared by this plastic, what you call the pipettes, and this is rubber teat, and this portion is the cylinder, what you call centrifuge tube, plastic centrifuge tube. So this is your dry powder inhaler. We have pumped the nanoparticulate or fumigated here inside, and the nose-only apparatus through the nose they have taken, and then we have investigated. Yeah, the nanoparticulate matter, both SEM and TEM pictures are there. You see here inside the TEM, you see drug particles are there inside. Here you can also see it. These are the TEM picture. And then the focus, I want to do it here, that this is your 30 minutes, 60 minutes, and 240 minutes. And this is, technetium level nanoparticles and free drug. This is technetium level nanoparticles, animals, and this is free drug animals. See, lungs, how changes is there. In 30 minutes, drug is they eliminating, whereas it is not eliminating. And see, the elimination is much more in 60 minutes and even much more in 240 minutes from free drug. But nanoparticulate drug, you see, even in 240 minutes, no much excretion is there. That indicates, it will remain in the lungs for longer time if we can deliver. This is a small portion of this entire study I have presented, just to make you understand that this is also another approach to target drug in the lungs without direct approaching. This is another drug targeting approach. I want to quickly finish because you may have questions in mind to discuss. So you see, here we have developed nanoliposomes. Nanoliposomes are, they are not particles, solid material. They are like balloon, inside is hollow, and membrane is there. So this is, very uniformly prepared nanoliposome, 30 nanometer to 40 nanometer in size. And then we have given, and we wanted to see. You just remember this particular chemical, 1,2-diasteryl glycerol phosphatidyl ethanolamine. This we normally say it is DSPE. DSPE is very important because DSPE is present in our brain. When we have developed the formulation with normal phospholipids, and we found it is going to brain, but the amount going to brain is not sufficient enough. It is very less. So we were thinking how it can be done, and all of a sudden we thought that if we can give some, lipids which is present in brain, whether it will be successful, and it causes miracle. These are the formulation we have prepared, and they're the normal data of size. Now you see, these are the liposome. Liposome means what is important, that you have to show that their membrane is intact. This is scartum. So you see the membranes are intact in every case. There's no single one is there that membrane is deformed or not. There's a bilamellar liposome in such a small size, 30-40 nanometers. So it's a small size liposome, but intact. Then it is leveled with FITC, that is, FITC has been incorporated along with the lipids, and we found that it is reaching in the brain. So that means it is successfully going to the brain. I can give you another important data of it, that when it is reaching in the brain, you see, it initially we found that when it is normal phospholipid, it is not going to brain. It is going to brain, but very, very less. Free drug, it is not going to brain at all. But when it is, phospholipid, it is this normal phospholipid liposome, it is going to brain, but very, very less. It's a miracle is there if you compare when we have just given, when we have just added what you called is simply phospholipid, brain phospholipid, along with this normal phospholipid to prepare the formulation, it causes a huge impact, and you see huge amount of drug, nearly 400 times increase of the formulation in brain. So that is a great success, you can say, of our formulation. And then also quantified it in terms of brain and in terms of blood level. So you see, in both the cases, you can see in brain how much change is there, you can understand, and also blood as free drug and formulation is a huge changes there. That means it is also remaining longer time. So it will produce a sustained release effect. Possibly this one, my last presentation. This is another interesting thing, that nerve targeting, peripheral nerve targeting, so far, possibly this is the first ever study. It has a very long history. This study, we took nearly 10 years to give the save. Initially for 4 years, we were unsuccessful, and then eventually we made it successful. Actually, I want to briefly tell you the story. Once I went to the what you call this Belagata ID, they have a scientific presentation. I presented one our nanoformulation related presentation. One doctor, senior doctor, he came. He said that I'm very impressed with this. So I want to just request you, can you just prepare some formulation loaded with ribavirin so that it can go to the peripheral nerve? So I said, we have done with central nervous system, but peripheral nervous system I have never done. But since you are saying, because he was working in the rabies area, anti-rabies, so he said that this drug in the in vitro laboratory scale, it works very nicely against the rabies virus, but in the system, since the rabies virus remains in the peripheral nerve, we cannot make it effective. So if you can bring it to the peripheral nerve, it will be effective. Now what happens, we took, I said, more than 10 years really to complete this study. What we did, first of all, our approach was to ligate it with procaine. Procaine, it will go to the peripheral nerve system. So obviously, we have used it. We tagged with the formulation, and then liposome. So it is tagging with the liposome surface, and actually, this is the what you call cartoon image of this liposome, and then we have injected it, and then we've gone for this study. So this is the chemical reactions we have produced, and thereby we conjugated it, And then we have studied it, confirmed it with this FTIR and then mass. And then once it is confirmed that conjugation is perfect, then we gone for this formulation related studies, discard FM and this normal size distribution, etc. Same. And then it's drug release also. And finally, what we did, we have gone for this technetium leveled formulation, and we wanted to see the presence of it in the peripheral nerve. It is very difficult to see because peripheral nerve, where can you see? So we have targeted the muscles area, see, and you see it is reached in the muscle area. Muscle area means the peripheral nerve area. So this is one document, but it is not substantive. You cannot say with this one document that yes, it is reaching to the peripheral nerve. For that, we have also gone for electromyogram, and then electromyogram, if you just compare that with this two study, you see here in normal, this is the electromyogram picture, and you see the graphics is this size changes because of the presence of procainamide. That indicates it reached in the peripheral nerve. This is the peripheral nerve impulse actually you have taken. So because of the presence of procainamide, peripheral nerve impulses change. That clearly indicates that yes, the formulation is reaching to the peripheral nerve. So far I have tried to give you brief description of different formulation, different approaches of drug targeting, and you know that if we can really target the formulation, because that is badly wanted, we can destroy the or we can control the destruction of our health problem related to misuse of drugs. Because whatever we are taking, we take normally the drug for a particular target, but it is distributed throughout the body, which is very harmful for our system. I think we have some time for discussion. Thank you very much. Thank you all for your listening. This is our university. This right side here, it is our department. I think those who are from Calcutta, they are well aware. And this is the stamp produced by the government of India related to Jadavpur University on its 50th year. Thank you very much. Thank you all.
Thank you so much, sir. This talk is very helpful for me, for our colleague, and as well as our participants. This time is open for the question and answer session. Sir, please give me the permission to share those questions which is available in the chat box.
I request you, I'm not very equipped with the computer, so somebody should read or somebody should
Yes, I will. Yes, I will read it.
I will try to answer. Yes, no problem.
Sir, first question is there any special criteria of a polymer or a drug to deliver to the ocular route by using nanosystem?
Ocular route is very, very sensitive route. Ocular route is not like other route. Although many people are working with this ocular route, there are two different problems. One is there is a very different balance problem, what you call the pressure problem. There is a different pressure problem with these two different anterior and posterior chambers of this eye. And secondly, so obviously, if you give something, the discharge of this ocular route is possibly much more as compared to the others, because the system is prepared like that. So ocular route, a very sensitive route also. I do not know for drug delivery through ocular route with nanoparticle, very difficult. But maybe liposome could be thought of, but still it may not be very comfortable, because this is a very complicated route. This is not like other routes. There major problem is, as I said, the two chambers has two different pressure balance. So that, and these two chambers has different concentration of this fluid, aqueous fluid and vitreous fluid. So that is a big issue. And that's why, whenever you will give something on the eyes, there is a possibility of discharge. Eyes never like to keep it on it. It wants to remove it at the earliest. That is a major problem.
Yes,
So
sir.
particulate matter may not be very successful. It will cause irritation. Obviously, I believe that maybe liposome could be considered, but still it is not very, attractive.
Okay, sir. Thank you. Sir, another question is, for any nano formulation with Taxol, how to select appropriate polymer?
See, whenever you are working with any drug, you first of all, your target should be where you want to apply, in which part of the body. So if you like to apply orally, or if you like to apply in vivo in the intravenous route or other route, so accordingly, because it depends on the selection of the polymers, whether it is biodegradable or may not be degradable. Like oral route, it may not be degradable. You can take it, but for like ethyl cellulose, you can consider it for oral route, but you cannot consider ethyl cellulose for the intravenous route because it is not degradable. So you have to consider the biodegradable polymer for intravenous, intramuscular routes, or when you are giving parenteral route, by parenteral route. But for oral route, it could be, you can select for this other polymers also. Second thing is, once you select the polymers, then first job will be the drug polymer interaction. You have to see whether drug is interacting with the polymer or not. That could be done with simply FTIR, DSC, or even IR or any other method. You have to check whether drug is interacting with the polymer or not. If it does not interact with the polymer, you can proceed for other selection with other excipients and all these things, and then you have to prepare the formulation, and then again you have to study the interaction, whether in the formulation stage interaction is physical or chemical. If it is a physical interaction, fine. But if it's a chemical interaction, you have to discard it again. So it's a kind of trial and errors. Basically, you have to take care of interaction pattern between these drugs and polymers, and there should not be any chemical interaction.
Thank you, sir. Sir, another question is, what is the role of surface charge in the drug delivery system?
It has enormous role. Basically, when the formulation, before administering the formulation, it has huge role. Surface charge, if it is greater than 30, plus minus 30, millivolt, what happens, it will remain in a suspended condition. So it will be easier for you to inject. But if it is less than that, you have to reconstitute it. You cannot prepare it for longer time and then inject. It will be particles will settle down. In those cases, you have to just prepare the suspension and inject at the earliest, otherwise it will choke your system. So this is a major problem, particularly before administering a drug in the system.
Okay, sir, last question is, what is the opinion if we use locked nucleic acid aptamer
I beg your
for
pardon?
Locked nucleic acid aptamer for this type of study? Is it increase the stability? Can I repeat, sir?
I understand, but it is difficult to say whether it will increase stability or decrease stability. You see, it's our system. You have to understand that it is our system, and locked one may not be very good, because you see, you have to maintain a specific 3D structure. So if with the lock condition it maintains a specific 3D structure, and it can bind with the protein, fine, fantastic, there's no problem. But you see, you have to be very sure about that person. Otherwise, you see, it may not work. And in the system, which one will work efficiently until and unless you work with it, it is difficult to say. Because there is no question of, what you call this foretell or future prediction. You have to do it and you have to understand whether it is working or not.
Yes, sir,
Yes, of course, one more thing is nowadays this computer facilities are there, what you call this, yes, I forget this term, computer modeling. By this also you can pre-assess the thing, whether it will work or not. But still, even if you pre-assess, I am telling you, out of 100, maybe 4 or 5 will work properly.
Okay, there is no question, sir, in the chat box.
May I have a...
Yes, please.
Sir, good afternoon, sir.
Good afternoon.
Hope you are doing well in this pandemic. Sir, my question is, if both the cell lines, like whatever you used in your first study, like HEPG2 and HUH7, both are the hepatocellular carcinoma cell lines. They are almost a similar type of cell lines in terms of the receptor they are expressing. Like so, and then what is the rationality behind choosing both of them in the same study? If both of them are similar.
See, the rule is that you have to select at least 2 cancerous cells in vitro study and 1 normal cell. This is the rule. And 2 cancerous cells, depending on that whether you have some specific target or not. Specific target is it that there are varieties of HCC? No. There are varieties of breast cancers, you see, like triple negative, ER positive. So there a question of specific selections is important. But HCC there is no varieties of HCC. So normally in those cases, if you have varieties of HCC, you can select different types of cells, and you can also target specific type of HCC. But here there is no option is there. That's why you have chosen the 2 HCC cells and 2 normal cells. But if you have a target, different target, like which is there in case of breast cancer, as I said, there you can choose different cell lines as per your own target.
Okay, thank you, sir.
Good afternoon, sir.
Good afternoon.
Sir, I have one query.
Yes, please.
So as we know that determination of zeta potential gives the stability prediction of the nanoformulation. So is it mandatory required for determination of stability by accelerated stability study?
No. It is absolutely required. Accelerated stability gives different thing. Zeta potential gives different thing. You are confused. Zeta potential actually, when is in a dry state, it has no meaning. When you are giving in a when you are just suspending it, then the question of zeta potential arises. And the suspending condition, if zeta potential, little earlier I said, greater than plus minus 30 millivolt, it will remain in the suspension for longer time. Otherwise, they will settle down. So this stability is different. This stability is not destruction of the formulation. This is just stability in a suspended condition. But when you are talking about the stability study, that is related to the stability of the drug first. In the formulation, how long the drug will remain stable. Second thing also, of course, you can investigate the physical stability of the formulations also. But basically, stability study, we indicate the stability of the drug in a formulation along with the physical stability of the formulation. That is totally different. And here you see stability of the, what you call this, zeta potential provides different stability. That is stability related to the formulation in a suspended condition. Okay.
Okay, sir, got it. Thank you.
Dear participants, any more questions? Anyone from the audience, you want to ask something? If no, then we can proceed. Sir, in this virtual world, This is a small token of appreciation from all of us. Kindly accept this as our love and remember us.
Yeah, I do hereby accept it. Thank you very much. And even if you don't give it, your love and affections always are there with me. So there is no problem. But still, I accept it wholeheartedly. Thank you very much. It's my pleasure to accept it.
Thank you, sir.
Thank you. Thank you.
Thank you, sir.
Thank you, sir.
So with this, we have come to an end of the first half of the 5th day. Dear participants, we will be joining for our last session at 2 p.m. sharp with Padma-Shri Professor Ajay Kumar Rai. And after that, we will have a valedictory session at 3:30 p.m. So it's a request from all of us to participate in both the session. And thank you till then.
So thank you very much. I'm leaving also.